Okay, welcome everyone to our October 2024 version of Symbiota support group. Today is going to be a little bit different because it's not quite as technical. We're not going to be showing you like a specific tool. We're going to kind of be talking a little bit broader about how a lot of organizations kind of talk to one another and databases talk to one another. So hopefully this is a little bit helpful for you to kind of contextualize. But before we move on to that, we'll just make a couple of quick announcements. The first one is hopefully you saw an email from us about Symbiota 3.1. We have officially released the newest code base to 3.1, which is very exciting. And Lindsay's just put a link to our announcement and some more details about that in the chat. So we are in the process of the relatively slow rollout of 3.1 into all the portals that are hosted by the Symbiota Support Hub. Portals with 3.1 currently include CCH2, the Lichen portal, the Bryophyte portal, Guatemala portal, and the African plants portal. And we will reach out ahead of time to the communities that are going to get 3.1 soon, so you know kind of to look for an update, and then we'll let you know once that update has been made, because then you'll experience slightly different functionality. And depending on the portal, you might see a little bit of different styling. We'll be working closely with the portal managers to make sure that everything is still looks nice and according to the community's needs. But just so you know, some slight changes might be coming to portals near you in the future. So keep your eyes on your email is the underscore. We are also mid-campaign for the Consortium of Small Vertebrate Collections, also known as CSV call, which is actually going to undergo a name change. I don't know if that's been announced yet, but stay tuned. If you manage vertebrate collections, the CSV call is going to get a new name soon. But regardless, if you know of any folks who are need some place to manage or mobilize their vertebrate specimen collections, this is the portal for you. Or if you manage your collections already in here, please let us know. We'd love to help you out with some data cleaning and some data mobilization help. So just touch base. All right, so let's go ahead and jump right into our topic of the day. And I hope that you all, during this kind of presentation part, are thinking about the organizations that you were hoping that we would describe how they connect to Symbiota, because I have some that I know we talk to people about a lot, but there are tons of other organizations and databases and things out there that I might not have put in this presentation. That doesn't mean that we don't talk to them or think about them. It just means that I didn't put them in this presentation. So keep those in your mind and we'll talk about them afterwards. Okay, so how do Symbiota portals relate to the broader biodiversity data landscape? Symbiota portals are just one puzzle piece in this really broad landscape. There's a lot of people doing excellent work related to or very parallel to what Symbiota is doing. So I want to highlight some of that. So I'm going to go through a couple of main categories that at least I had in my mind when I was thinking about this topic. And then, like I said, we'll go to questions at the end. So one that I wanted to talk about, and that is most frequently discussed, is specimen databases. So database is a pretty broad word that can just mean like where you store data related to a thing. And in this case, I'm talking specifically about specimen data. So data about collected things that are now stored in natural history collections or other sample collections. And the way I was thinking about this is there's kind of a spectrum on the line of collection management system to data aggregator. And the difference between these two extremes on the spectrum is that a collection management system is focused primarily on data editing and data providers. So it's focusing on the people who are entering the data, the people who have to enter annotations or manage loans or digitize. That's what collection management systems are mostly focused on. Whereas data aggregators, they are generally based on, or they're focused on data viewing. So usually multiple sources are going into a single aggregator. That's why it's called an aggregator. It's aggregating from lots of places. And the focus is more on data users in this case. So the people who are trying to visualize the data to make land management decisions, the people who are downloading the data to do research, and then any edits that happen in a data aggregator are usually just added on data. They don't go directly to the source because they're in an aggregator. So some examples on each side of these spectrum, and none is probably perfectly on one side or another. But one example or several examples of collection management systems would be things like EarthCape, Axial Emu, Specify, Collection Space, Microsoft Access. Like these are things that are databases that are generally managed locally. Sometimes they're installed like on the herbarium computer or on an institutional server. They're generally things that are managed by your institution or your collection. And then some examples of data aggregators include iDigBio, GBIF, VertNet, and the list goes on and on and on. There's a lot of places who are aggregating specimen data. Then kind of in this middle between aggregator and strict collection management system, you have a couple of other softwares and platforms. For example, Arctos. Arctos has a lot of collection management focus, but it also is a place where multiple collections are managing their data in the same place, and you could technically search across multiple collections. So that's why it's kind of here in the middle, because there's lots of data editing functions, but there's also data aggregating functions. The tricky thing about putting Symbiota on this axis is depending on the portal, Symbiota portals can lean anywhere from collection management systems to data aggregator. And that's why it's kind of hard to say like, well, Symbiota is kind of like GBIF, but it's kind of like Specify, but it's not really one or the other. It's in this middle ground. So we can think of, for example, right here in the middle might be a portal that has some snapshots and some live managed collections. By that, I mean, some collections use that Symbiota portal to enter their data, edit their data, do digitization directly in that portal. And that's the main place where they do that. But other collections might just put a copy of their specimen data in there so that it can be accessible and searchable by the public. So an example of that might be CCH2. So CCH2 is the Consortium of California Herbaria. And there are lots of collections that use it as their primary data management system. And then an example of more of a data aggregator would be the Pteridophyte Collections Consortium, also known as the Pterido Portal. And the reason for that is because just copies of the fern data coming from other sources are in the Pterido Portal. So people can people can search all of the fern data in one portal, but the actual management of the fern data happens in other Symbiota portals, in Specify, in Emu, a bunch of other places. So Symbiota portals are very flexible. They can be on this extreme data aggregator where everything's just a copy, or they can be on this extreme collection management system, because you could install a Symbiota portal on your laptop and then nobody could ever view it except for you because it's only on your laptop. So that would be the extreme version of only a collection management system that you use privately locally. But the reality is most of them exist somewhere in this middle ground spectrum where there are some snapshots, so just copies of the data, and some live managed collections, which are the live active databasing locations for those collections. Does that make sense? I know a lot of this is review for many people, but I want to make sure that we're all on the same page. Thumbs up, emojis. You can react in Zoom. Got a thumbs up from Lindsay, Chilla, Joe. Great. Okay, we're on the same page here. I just wanted to throw iNaturalist in here too. So iNaturalist, I would consider somewhere between observation management system and data aggregator. But again, they focus on observations, not necessarily specimens. Okay, so a critical part of kind of defining all these different types of collections is defining how these different types of organizations and databases talk to each other. How does data get from A to B, B to C, or between any of these organizations? Well, how these things relate to Symbiota is often things come from more specific collection management systems into a Symbiota portal to be in concert with any live managed data sets that are in that portal. And then oftentimes, a lot of the data goes directly to GBIF from a Symbiota portal. That being said, you can also have connectors that go from specify to GBIF, specify to VernNet, CSpace to iDigBio. Like, we're not the only conduit. This is just often, this is how data can go in and out of Symbiota, and also from Arctos. And because Symbiota portals adhere to Darwin Core standards, which means that we have kind of an easily translatable package of data that can go back and forth. That means that Symbiota portals can ingest data sets from aggregators like GBIF or iDigBio, and they can also send those packages of data to collection management systems. There is a caveat though. So with this spectrum from collection management system to data aggregator, there's also kind of an increasing amount of interoperability because you're mushing a bunch of specimen data sets together. So collection management systems tend to be less interoperable because they have lots of custom tables and references between tables that can be really difficult to map other people's data sets onto. So if you have something that is just a collection management system and it doesn't have any aggregation functions, then it can be pretty difficult to bring data into it. But then data aggregators, like that's their whole job, is to bring data into things. So they tend to be much more interoperable. So that's why there are these dashed lines going from Symbiota to Arctos and from Symbiota to other collection management systems, is because they have to be able to ingest this Darwin Core archive, aka this package of data that aligns with biodiversity standards, into their database, which depends on their system, not on any of the data aggregators' systems or Symbiota system. This has been a problem in previous digitization collaborations, because people will do some stuff in a Symbiota portal and then they'll just hand it back and be like, put this in your collection management system. And then that link that can't happen because of the limitations of the collection management system. So that's kind of been a trouble point in the past. I know that that is getting easier in some of these collection management systems. For example, I think that Specify is building a tool that enables them to bring partial data into their collection management system, but it's definitely not always easy to do that. So I could go into a little bit more detail about the ways that data can get into Symbiota portals, and I'll just kind of do this briefly. So Specify has two main ways of getting your data into a Symbiota portal. One is called the Symbiota Specify Connector, and I believe this only exists in Specify 6. So it's not super well supported anymore because Specify is mostly focusing on developing and supporting Specify 7, which is a little bit more flashy and has more features. So that I wouldn't necessarily rely on, but a lot of people still use that to put their data into a Symbiota portal. The second way is to make a Darwin Core compliant package of your data available via an RSS feed to a Symbiota portal. And then the Symbiota manager or you can hop into that RSS feed and copy the link to your Darwin Core archive and import it into the Symbiota portal. So if you're working in Specify, those are the two main ways that you could get your data into a Symbiota portal. If you're working in EMU, you have to create a custom report, I believe is what it's called. So you have to kind of write some scripts that are going to put your data into a Darwin Core formatted thing. I don't know very many details about how to do that, but I know that people in the community are doing that. So reach out to them to get more info about that. And then I don't know too much about EarthCaping collection space, but I'm sure there's some sort of exporting function that you could use to create at least a CSV file, because Symbiota portals can import CSV files. Another way that all of these collections sometimes get their data into a Symbiota portal is via what's called an Integrated Publishing Toolkit, or an IPT. This is a software that GBIF has developed that kind of allows people to map their data into Darwin Core and then get it pushed to GBIF. Because that mapping and that packaging happens in the IPT, Symbiota portals can also grab that same link that GBIF harvests and pull that into the Symbiota portal. So that data transfer can happen. And then to go the opposite direction, to go from an aggregator into a Symbiota portal is very easy because it's very easy to download a Darwin Core archive from any of these aggregators. You just download it, you use the Darwin Core import tool, and it's done. So one important note about all of these processes is that none of them are automated. There might be some automations between these folks that I don't know of, but in terms of getting data into or out of a Symbiota portal, there are currently no automated processes. So people will often come to me and they're like, I thought our data was automatically updated in iDigBio. That's actually not the case. Your data has to be manually triggered to be updated by GBIF. And then it has to be manually triggered on iDigBio's and to be updated in iDigBio. And I'll go into a little bit more detail about that in this next section. But first I want to pause and take a breath and see if anyone has any questions specifically about specimen data flow. Oh, and one thing I didn't mention is how iNaturalist data gets into Symbiota portals. That's usually downloading a Darwin Core archive from iNaturalist and then just uploading it into the portal. I don't think we have any sort of API access to iNaturalist observations. Mark, you have a question.
Yeah, kind of on the topic that you just mentioned, APIs. My understanding is that there is an API that's under development for Symbiota, but is not in production yet on any of the portals. Is that the case?
That is correct. Yeah, it has some like very basic features built out where you could do some querying, but we haven't written up any documentation and it's not fully fledged. Any other questions?
Could you say the name again of the tool that GBIF uses to format?
Yeah, it's called an integrated publishing toolkit, and it's a software that you download and configure on your own server and then map your data.
Thank you.
And then Lindsay answered this in the chat, but I'll go ahead and ask it openly. Karen asks, does data flow from one portal to another, for example, the consortium of bryophytes to the consortium of Pacific Northwest herbaria? Great question on two accounts. The consortium of Pacific Northwest herbaria is actually not a Symbiota portal. It's just another portal software that was developed, I believe, by folks at University of, I don't want to say wrong, but British Columbia, or maybe, I think it's British Columbia. No, Washington.
I thought
Mark
they
was,
I thought they just made a new Symbiota portal. But maybe
I
not.
don't know anything about that, but it's possible.
Okay, thanks.
But then data do get moved between different portals, and I will kind of show an example of that. To go back to the Teredo portal example, most of the collections in the Teredo portal come from another Symbiota portal. So this is just a database of only the fern data coming from a bunch of sources, but many of those being Symbiota portals. So if I click this More Info button, you can see that the data is coming from the IND collection in the Consortium of Midwest Herbaria portal. And it's giving me a link that's just going to help me download the Darwin Core Archive, so I'm not going to necessarily do that. But Midwest herbaria. These data sets are coming, for example, from the Midwest herbarium, or if I have something coming from California, it's probably coming from the Consortium of California herbaria. For example, the California Botanic Garden. And these are also not automated, meaning they do have to be triggered, but they are already set up. So because stuff can pass so easily as a Darwin Core archive from one portal to another, and using some other built-in tools, we can just pass the data from one database to the next database after we trigger whatever needs to be triggered. And this happens for most Symbiota Support Hub hosted portals on the first week of the month. Thanks to iDigBio for our portal maintenance funds that we have through them. And also individual funders have been able to say, hey, we're supporting this particular portal, and that's enabled us to have the bandwidth to update all these snapshots monthly. But again, it's not something that happens necessarily automatically. And Mark clarifies that the UBC cannot take credit for the Pacific Northwest herbaria.org people down at the Burke at the University of Washington. Okay, thanks for clarifying, Mark. We do integrate some data from the Consortium of Pacific Northwest herbaria because they make their data available as Darwin Core archives too. For example, the Portland State University herbarium shares their data with, I believe, the bryophyte and maybe also the lichen portal. So it's very easy to pass data between those databases. Other questions? All right, let's hop back in then. Feel free to throw questions in the chat as we go, and I'll address them as we go too. Okay, I wanted to hone in on a couple of key relationships that we have, and one of them is between Symbiota and GBIF. And this is kind of on two levels. I'm going to talk about it on more like an organizational level first, and then I'll talk about it on more of a database level. So GBIF is an intergovernmental organization that has nodes of different countries that join that node, and they pay in and support this great database and data aggregation organization that really helps mobilize not only specimen data, but just occurrence data relating to natural history and organisms. But then there are also nodes that are organizations that are not governments. And the Symbiota Support Hub is one of those nodes. Just like VertNet is one of those nodes, the Biodiversity Heritage Library is one of those nodes, etc. We're just kind of accessory organizations that agree with the mission of GBIF to mobilize data, and we try our best to do that. I mean, that's part of our mission too, and it's very synergistic with GBIF. So that's kind of us on an organizational level. We have the ability to endorse publishers, which helps you guys get your data out into GBIF faster. And Symbiota portals also have built-in publishing tools that enable us to push the data from our portals directly into GBIF. So usually this is done with collections that manage their data live in a Symbiota portal, but snapshots can also publish from a Symbiota portal to GBIF, as long as they're not also doing that in some other manner. And this usually means that one Symbiota collection becomes a data set in GBIF. So what do I mean by those? A collection is one entity that you're managing in a Symbiota portal. So this is my collection in the CCH2 portal. But if I open up the bryophyte collections, I have another collection because it's coming from a different portal. So this is considered a separate collection in like the Symbiota world. Even though they might be in the same room, they are stored in a different collection in a different portal. So when I publish them to GBIF, each collection is going to become its own data set. And what happens is the data from this collection, specifically like the description and the contacts, this gets ported over to the data set in GBIF. So this description matches my collection. This is my vascular plant collection. I say my, but it's the one that I'm affiliated with. Jen Yost is the director. But then if I go kind of one level up in the hierarchy, there's this California Polytechnic, San Luis Obispo. This is the publisher, the organization that owns all of these data sets. And this description actually has to be managed by GBIF. So if you see any issues with your publisher descriptions, then you'll have to talk to GBIF about those because they're not actually updated from your data sets. I just said this, your Symbiota collection description becomes your GBIF dataset description. And then your GBIF publisher description is not linked to your Symbiota collection description because your publisher is the organization kind of above each data set and linking all those data sets together. So your data set that's coming from Bryophyte portal, from Lichen portal, from Myco portal, from Algae portal, what have you, all those can be data sets underneath your one publisher. Again, this process of publishing your data to GBIF is not automated, but it's very easy to trigger once it's been set up. And the setup is actually very easy too. The hardest part is basically registering for an organization under GBIF. And that's still not hard, it's just kind of more bureaucratic. So like I could go to my collection of bryophytes, which is linked to a GBIF collection. go to my Darwin Core Archive publishing and say refresh Darwin Core Archive, and it says starting GBIF harvest. Let me try to zoom in here. Triggering crawl, so that's going out saying GBIF, I have new data, and then GBIF grabs the new data, starts its crawl, and brings in the new data, usually within like 15 minutes to a half an hour, depending on how backed up they are. And that's how easy it is to publish to GBIF and to update your data in GBIF. Any questions about that? Harry says a poster or PDF of all these entities, what they are and how they're related would be nice for a reference. Are the PowerPoint slides available as a PDF? We could certainly make them available, Terry. Yeah, that's a good idea. I know that there's a group called the International Partners for the Digital Extended Specimen, of which we are also a part, who are kind of working on how they can show this grand network talking to each other. But it's also much broader than what I'm presenting right here. Donna asks, what is an RSS feed? I will try to give you a definition, but my understanding is also kind of like a little bit high level. It's basically like a running feed in the computer world that computers can see once they're updated, and that will trigger them to pull more data or pull the information that exists. Mark could probably give a better description, but I don't need to put you on the spot if you don't want to.
Yeah, for people that were in the early internet days, it was really commonly used by blogs. It's basically a way for a system to just broadcast information on a continuous basis, and it does it just via a text stream. So, like as blog posts go up, they would just get added to that stream, and then things would read it. The same thing happens for data sets that are available. Your instance is just broadcasting, "Hey, I have this available now. Come and get it."
Thanks, Mark.
Just a quick question. Well, I mean, is this anything like what it used to be called ITPs? I mean, back in the days of Veronica and Archie's, which is, I think, maybe even older than what Mark is talking about. Or it's just the way information moves around. We probably don't really need to understand it completely.
Mark's giving a thumbs up. Yeah, it's one of those things that I like understand in practice, but I couldn't tell you how to set up an RSS feed. Okay, any other questions? All right, moving right along, I'll talk about another major relationship that we have. Oh, there's a question. Allison asks, how often should we share to GBIF? After new data? Great question, Allison. I also refresh Darwin Core Archives for the whole portals because I have a way of triggering that for everyone who's currently publishing the first week of the month, along with all that snapshot updating. But if you ever do like a major upload, you upload a couple thousand specimen records or you upload several images, it wouldn't be a bad idea to go ahead and push those to GBIF because then they're going to be more available to a broader audience quicker. So up to you, especially since it's so easy. All right, another critical relationship is between Symbiota and iDigBio. So I'm going to talk about this again on the organizational level and then on the data level. So iDigBio is a US-based institution that, or organization that was funded by the National Science Foundation. And it's been around for about, 13-ish years, 12-ish years. And its purpose is to develop and enhance training, research, communication, and a lot of other things with regard to digitizing and managing biodiversity specimens. This is my short summary. You can learn more about their mission and vision on their website, but this is generally what they do is they're like a coordinating body that helps get energy into where digitization needs to progress, needs to happen. And so they provide workshops and trainings, etc. They also have a data portal, which is strictly an aggregator for US-based specimen data. So specimens that come from US institutions. And only specimens, not observations. So that's kind of the overlap of GBIF and iDigBio, is that GBIF is way bigger than iDigBio because it's just US-based specimens. The Symbiota Support Hub currently gets part of our funding from iDigBio. So we are a domain of iDigBio, which just means one of the functional parts of iDigBio, because we got some funding through the National Science Foundation to become the Symbiota Support Hub. This is the current what we call the Symbiota Support Hub. But the positions that were funded by iDigBio are these 3 positions. It's Lindsay, Greg, and me, with some additional funding for our PIs. And so if you're a little bit confused about like, wait, is like Symbiota part of iDigBio? Does Symbiota get all of its funding through iDigBio? Is Symbiota going to last beyond iDigBio? Oh no, they're ending in 2026. Don't worry, we actually have a little more of a diversified funding stream. The funds for our PIs and our developers actually come from many different sources, including other awards and contracts. So that's the organizational side, is we're part of iDigBio, but also kind of, if you think about this circle having like a blob on the outside too, we're also kind of extra iDigBio on some level too. So data can go from Symbiota portals into iDigBio, but that is also not an automated process. I think the current workflow is you just email iDigBio and say like, please put this in your refresh feed. I'm not sure what their refresh rate is without you manually asking them to. I want to say it's like biannually. But I know that they've had some IT personnel changes, so I can't speak necessarily for them. Okay, any questions about iDigBio? All right, moving right along. I want to make just a couple of comments about a couple of other acronyms and organizations you might see flying around in the biodiversity specimen ecosystem. I want to talk about non-specimen databases. One of those could be considered taxonomic databases. So those are databases of scientific names and relationships between those names and synonymies of those names. So I'm thinking of things like the Catalog of Life, which has kind of as its serving platform this thing called Checklist Bank. There's the World Register of Marine Species, there's Tropicos, there's Index Fungorum, there's Plants of the World Online. There's a long list of taxonomic databases from really large, like Catalog of Life, which covers all taxonomy, theoretically. And there's smaller groups, like there's an ant taxonomy database, and there's just smaller groups of taxonomic databases. So how do these connect to Symbiota portals? Well, the ones that are listed here with a little asterisk on the Tropicos that I'll talk about in a second actually have direct linkages into Symbiota portals. Those direct linkages are via the taxonomic cleaning tool. So if you ever run the taxonomic cleaning tool against your dataset, it's going to look in whatever the selected taxonomic database is for any names that aren't already in the taxonomic thesaurus of your portal. And if it finds that name, it already knows how to harvest the data associated with that name, like its a higher taxonomy, its synonyms, things like that. It's going to pull them in. For example, it looked for Rosa borboniana, and it said, I found that. It's actually listed as a synonym of Rosa indica. So I brought in Rosa indica and I brought in Rosa borboniana. And so it linked those all together. And as I mentioned, we have linkages to the Catalog of Life. That's through the Checklist Bank API. API just means Application Programming Interface. It means a thing that enables computers to talk to each other, basically, and pass data back and forth. We also have a linkage to the World Register of Marine Species. We also have one to Index Fungorum through an API called FDEX. And then we did have one to Tropicos as well. However, Tropicos didn't pass us synonymy data as well. So it ended up bringing in everything as an accepted name, which kind of made a mess. So generally, we don't enable that Tropicos API anymore. Okay, and an important clarification about the relationships between the taxonomic databases and the Symbiota databases is that Symbiota portals can harvest new names and their synonymy from some taxonomic databases, but there is no automated taxonomic synchronization after this initial upload. So your taxonomic thesaurus is not going to automatically update with newest changes in Checklist Bank or Tropicos or Index Fungorum. And it can become stale because it's not linked up to those automatic updates. That's more of a manual process that needs to happen afterwards. We're aware that it would be awesome to be able to sync up databases, but that's a pretty huge lift that we haven't been able to get any funding for or dedicate any of our time to. So if you're thinking of ways that you could have better taxonomic synchronization, write us into your next NSF proposal because we'd love to be able to update taxonomies very frequently. Al makes the point that Catalog of Life botanical names aren't always consistent with Jepson for California plants. Yeah, totally, Al. There are, especially in kind of botanically, like super botanically active places like California and the Southeast United States, there are like local floras that people prefer over the big aggregator ones that we can hook into. And so in those cases, often the portal managers or some specified folks will kind of be tasked with going and updating names as necessary. Like in the past, I have physically gone into the Jepson updates and made sure that any of the new names were in there and made sure that any of the synonymy has been switched. But again, this is a pretty manual process. And Jason Alexander has worked on that in California too. John asks, are there plans to incorporate Eschmeyer's catalog of fishes into the taxonomic cleaning tool? Not to my knowledge. I don't know if they have an API, which is kind of what is required to build up that type of tool, but that could be a cool integration for future development too. Oh, thanks for sharing that link, John. Any other questions about taxonomic integrations?
I might mention that I'm not surprised that you had trouble with Tropicos because they don't indicate what they think is the acceptable name.
Yeah, exactly. It's like you can bring in new names, but you can't indicate whether it should be one thing or another.
You might look at Q Plants of the World. They do make a decision.
Yeah,
acceptable.
and I think they have an API too. So it theoretically could be integrated into the taxonomic cleaning tool. It's just one of those many things that we wish that we had time to do. Karen asks, do we use Brio names? I know that there has been some synchronization work between Brio names and the Bryophyte portal, thanks to the Global Thematic Collections Network, which was a lichen and Bryophyte digitization project. But no, it wasn't linked directly via the taxonomic cleaning tool at that point. Allison says, I have simply been overriding the pop-up warning when I enter a name not in the thesaurus and typing in the name or authority as written on the specimen anyway. Is that an okay to proceed? Yep, absolutely. A name does not have to be in the taxonomic thesaurus in order for it to show up in your portal. I would recommend, since you probably are adding newish names, since you're doing the African digitization, I would probably run the taxonomic cleaning tool relatively frequently so that they can pull any new names that are in Catalog of Life into the taxonomic thesaurus, and you can improve it for everyone. All right, just a couple more combos to talk about. There are also databases of organizations. The two main ones that I'm aware of is the Global Registry of Scientific Collections and also Index Herbariorum. The Global Registry of Scientific Collections is this snazzy portal that has a, as it mentions, a global registry of scientific collections that it knows about. And those can be institutions or they can be individual collections. You can kind of think of these as being like the GBIF publishers versus the data sets, because often these collections are the fungal collection is curated separately than the insect collection, curated separately than the vascular plants, things like that. And I don't have time to go too far into detail, but this exists irrespective of the data that is published to GBIF. So you probably exist in here, even if you have not published your data into GBIF. Once you have published your data into GBIF, you can link it to your entry in this global registry to say like, hey, here's information about my collection, and here's where you can find our data. But this is not quite the same thing as like a database of data. This is a database of collections. And then the Index Herbariorum is a database of specifically herbaria, so plant collections, and probably also some fungal collections are listed in here, belonging to or existing in the world. And Index Herbariorum and the Global Registry of Scientific Collections, they all talk to each other. So anything that goes in Index Herbariorum eventually makes it into the GR cycle, as we call this. And the reason why this relates to Symbiota is because you can think of your institution as your larger collection and then of your larger organization. And then each Symbiota collection you have can be a collection in here, or it can be one collection in Symbiota can actually be linked to multiple collections in GR cycle. So this is just another database where information about your collection can exist and it can be useful to you. We should probably have a Symbiota support group just about GR cycle in the future because there's some cool new dashboard tools that enable you like visualize your data. So we'll talk about that in the future. And this is just a visual diagram of GR cycle is real big, and then Index Herbariorum just contains herbarium data. Okay, two other non-data organizations that you'll hear about frequently in our sphere would be the Biodiversity Information Standards Group, also known as TADWIG. That's because they used to have a different acronym and everybody called them TADWIG, and then they changed their name, but everyone still knew them as TADWIG. So they're still kind of TADWIG. But their main relationship to Symbiota is that they made and they maintain Darwin Core and a bunch of other standards. So when I talk about a Darwin Core archive, that's a package of data using the particular terms and recommendations by this group in terms of Darwin Core. And so many conversations in this organization actually directly form how we develop the Symbiota database because they're doing a lot of thinking about how concepts should relate to one another. And that's actually really useful thinking to inform how the data should be structured. It's not one-to-one, just so you know that these folks are out there doing biodiversity standards work, and we're listening closely into what's going on in that realm. And then another group that we interact with frequently is the Society for the Preservation of Natural History Collections, for obvious reasons that they have natural history collections and we have tools that support natural history collections. So that's kind of that. Okay, I didn't leave too much time for questions, but I do want to know what organizations or acronyms are puzzling you. What do you want to know more about? And I'm going to look in the chat real quick. Okay, any other questions? Things I didn't touch on? Organizations you're trying to fit into the puzzle?
I have a question, and I tuned in late, so I might have missed this, but I'd like to know, like, why GBIF. My impression is that servers in the US can see our portals here, but if we want people the rest of the world to be able to access our data, it has to be reserved, aggregated and reserved through GBIF, or is that not correct?
Not necessarily. There could be limitations based on certain countries that have like sanctions against the US that would disable them from viewing things on US servers, but I haven't heard too much about that. But no, anyone with internet access and who are able to access websites on US servers, which should be most of the world, should be able to access the Symbiota portal just fine.
So then why do we also share data with GBIF? What do they do that we can't?
They do a lot of things, and I can link to a recording for you. But one of them is that they track citations a lot better than we do. We don't track citations at all. We just don't have that kind of infrastructure. But kind of the main reason is that like they have a much bigger portal that's known by a much bigger community of people. And so our data is aligned with standards and able to be downloaded with tons of other data, and that can enable more powerful research because they don't have to specifically look for a California specific portal because maybe they didn't know that their genus had endemic species in California and they're doing a big search on one genus, for example.
Okay, thanks.
No problem.
Katie, I'm somewhat confused by, and this is, I guess, an iDigBio question really, is when they say they serve US data or it's a US portal, does that mean, so for example, White WVU Herbarium has collections from all over the world. There are foreign herbaria that have collections from the US. And so is iDigBio focused on the institutional home being in the US and then they're interested in all my data regardless, or do they only want collection site localities in the United States?
The first one. They mobilize data from US-based institutions. And I think the reasoning behind that is that all of these thematic collections networks were funded, then they were funding US institutions, and they needed places to mobilize that data to. And so iDigBio's mandate was, we're going to make a portal that's going to enable all that new data to go out. Symbiota portals were like developing kind of in parallel as both collection management and aggregators. So the need for that is, I mean, maybe there's still a need for that, but that was iDigBio's main mission.
To me, that links with Allison's question that why would I have a larger interest in GBIF if I'm interested in the flora of West Virginia? There's no reason that somehow some, collection might be in an African, European, Asian herbarium, and why wouldn't I like to know about that? So it's just that GBIF again gives a broader view.
Totally. Yeah. Yeah, there are definitely a lot of non-US institutions that have US specimens. We all send our explorers everywhere, kind of. Other questions? I see we're right at the hour, but I don't want to cut off anyone if they want some clarification. All right, seems like we're good. Thanks for tuning in, everyone. I hope today was a little bit useful in filling in some missing pieces. Feel free to send any questions, follow-ups to us. Or if you have suggestions for future Symbiota support group meetings, we're totally open to that and happy to cover whatever topics are most pressing to you. Thanks, everyone. Have a great rest of your week.